see and drop trace harm plants. This trace reduce plant growth, close stomata, increase salt, I mean sodium inside the plant, and lower photosynthesis. Roots are also affected because they absorb less water and nutrients and produce harmful molecules like ROS. But some helpful bacteria like PGPR, I mean the plant growth promoting rhizobacteria, they can protect the plants. In the upper plant, they help to improve the water movement, keep the soil level balanced, and slow down aging. In the roots, they can help absorb nutrients, maintain the hormone balance, reduce stress, and produce useful enzyme and chemicals. So PGPR play an important role to help plants to survive in tough conditions.
Ma'am, kindly
Now,
conclude. Yeah, ma'am, kindly conclude, ma'am. Hello. Hello.
yes, sir.
Yeah, ma'am, kindly conclude, ma'am, your presentation. Kindly conclude.
Okay.
Okay. Then.
So I have discussed about some solutions like some biotechnical solution and what policies we can take and how further research can make the things better. So, in conclusion, we can tell that climate change threatens plant microbe interaction, impacting the plant health and agriculture. However, utilizing beneficial microbes can help create resistant ecosystem. A combined approach of the research policy and regenerative farming, and by the help of some biotechnical techniques, we can ensure the food security and environment stability. And these are some references because this was a literature review. So these references help me to gather the information. And thank you.
Thank you so much, ma'am. It was a nice presentation regarding the dynamics and implication of climate-driven disruption in plant microbe interaction. So we are now open to the interactive session, and I will request the participants to ask something if they have. Uh, Dr. Ashok
Sir,
Bhattacharya, sir, do
is
you
my
want to
screen
ask?
is still sir, is my
Yeah,
screen is still showing? Or
yeah,
it
yeah,
has
yeah.
stopped?
Your presentation is not visible now.
Okay, okay.
So I will request Dr. Ashok Bhattacharya, sir, if he has any question. So yeah, you have raised a hand. You have any questions, sir? Okay. Dr. Prashanto Shah has a question, so I will request Dr. Prashanto Shah if you have any question.
So am I audible?
Yes, sir, you
Am
are audible, sir.
I audible, Dr.
Yes, yes, you are audible,
Bhattacharya?
sir.
Okay, fine.
Yeah, yeah, yeah.
Right. Okay, I just have a very elementary question to ask. That is, you talked about the salinity remediation or mitigation by the microbial effect, right? So I would
Yes,
just
sir.
like to understand whether the microbes, the PGPRs that you are talking about, they were basically the rhizoplane based or the rhizosphere based?
Sir, I just fear based.
Rhizosphere means since you told about certain mechanisms,
Yes.
so that's why I was intending to know. So you're confirming it is the rhizosphere based, right?
Sir, actually, I have done a review work. So the papers I have read, it was Rajasthan based. Actually, I am not
Okay,
aware that is there any activity in Rajasthan based. Or,
okay. Okay, fine, fine.
So I will request the other participants if they have any question. Okay, so I think no questions are there. So thank you so much, Shomita ma'am, for your presentation. And I will now request Dr. Sohardo Ray to present his presentation that is regarding the roots of healing, a review of bridging traditional plant wisdom to modern medicine. Sir, are you there?
Am I audible?
Yeah, you are audible.
Sir,
Sir,
I am
do you
not
want us?
doctorate. I am not doctorate. I am normally I
Okay,
have not
okay, okay,
completed my thesis, so I am not doctorate.
okay.
So
Okay, so
uh,
sir, you want us to present your slide or you will share it by yourself?
sir, sir, I tried. If not happen, then you just
Sure, 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 sure.
Is my slide visible?
Yeah, yeah. Yeah, your slide is visible, and after that, I will request Dr. Miss Songita Mondol to prepare herself. Yeah,
So slide is
Mr.
visible?
Yeah, your slide is visible, Mr. Sohardo Rai.
Yes, sir.
You can start now.
So, yes. So good afternoon, everyone. Myself, Shobhar Rai. I am guest lecturer for Department of Botany, Bahrampur Girls College. And today my topic is Roots of Healing, a review of bridging traditional plant wisdom to modern medicine. So basically it's a review paper, and the aims and the objective of my review paper is mainly based on the in previously there are many traditional plant which are used in medicinal purposes, and these plants are again used as pharmaceutical development, and as well there are many implants in secondary metabolites are also present. So basically using analytic techniques and genetical studies, we have to study the plant adaptability in medicinal potential and highlight the importance of biodiversity conservation and preserving the traditional medicinal plants for supporting the global medicinal advancement. So we all know in previous time, and based on it decades or more than a decades, there are many plant-based medicine which are used in human health. And now the modern recognition is required for giving them and acknowledge them the and acknowledge the value of the ancestral remedies. So basically, there are many scientific validation is already done via phytochemical analysis, like from willow bark, aspirin is prepared, and so on. And not only this, the molecular biology and genetical studies have revealed the practice and the mechanisms. So the study mainly based on few steps. First of all, the literature review, as it is a review paper or based on review research paper. So first of all, many review by academic journals have studied. After that, not only journals but also various database and studies, and from there various phytochemical analysis were found. After that, the phytochemicals found are clearly or visible, so they are tested in laboratory, and proper database and analytic techniques, chromatography and spectroscopy type techniques are used for verify it. And not only verify, but also ethnobotanical surveys are done, and check the how kind of medicines are used by the tribal people mainly here, and check the phytochemicals present, and other ethical aspects are also followed. So the results find out that the phytochemical confirmation, there are many kinds of phytochemicals found, like
Sir, your slides are not
tannin.
changing. Hello? Sir,
Yes, sir.
your slides are not changing.
Sir, slides are not changing.
No, no, no.
Is
Yeah,
it
now it
changing
is.
now?
No, yeah, now it is. Now it is. Yeah. Uh, Now your slides are not visible, sir. Do you want us to control by ourselves?
Yes, sir, please.
Okay, okay, fine. Wait, please wait.
Yes, sir.
Okay, wait. Please wait. Please wait for 2 minutes.
Yes, sir.
Yeah. So, is my screen visible now to everyone? Kindly reply.
Yes, sir. Yes, it's visible.
Yeah, it is visible. Fine. Okay. So here is the slide, and kindly tell me then from which slide I will start.
Sir, next slide. Please go next slide.
Okay.
Next.
Yeah.
Uh, I have already covered this. So if you
Okay.
tell our participants tell to recap, so then I will recap. Otherwise, please next
No,
slide.
no. You can continue from your cursor.
Okay. So next slide.
Yeah.
Next slide. Next slide. Yeah. So after the methodology I have mentioned, and from this
Mhm.
methodology, phytochemical conformations have performed mainly the
Yeah.
secondary metabolite stage, like, and not only secondary metabolites but also the quantitative and qualitative performance have performed. and after that the ethical consideration and sustainability work. And this work mainly is a try to create a bridge between the traditional plant to the modern studies and give a create a collaborative model between the tribals and the researchers, and create a benefit sharing, right, mutualism. Next slide. So here, this is the test of secondary metabolites which I have performed. Like here are alkaloid test, protein test, carbohydrate test in different concentration, and other ethanol extract and acetone extract have followed. Next slide. So from these studies, we have clearly visible that the in previously the knowledge the ancient people use is clearly scientific knowledge, and which can be utilized in regarding the today's life is very much. And this in this way a interdisciplinary research can be provided, and in future at it various kind of drug discovery and scientific studies can be performed. Next slide. So from this study, I made a conclusion that the traditional science is can be a helpful tool for modern science, and the knowledge they have, the benefit they have, they must be give the justice and give them the priority and their knowledge as well, and create a collaborative framework between ethnobotanists and the recent studies, and going to toward a future research from this knowledge. Next slide. So the here are various challenges coming with, like uh, current people can be not supportive the document, their culture, but still we have come to a way like standardize a phytochemical protocol, development a comprehensive genome database for medicinal plant, and establish international argument to protect them. Next slide, please. So I am very much thankful to Department of Botany, Durgapur Government College for allowing me to present this. And I am very much thankful for the people who helped me throughout the studies. Next slide. Here are the selective references for the study. Next slide. Thank you so much.
Thank you so much, sir, for your nice presentation. So now we are open for interactive session. So I will request our participants if they have any question. Sir, can I stop presenting?
Yes, sir.
Okay, okay,
Yes, sir. I have in my side.
okay, okay.
Any questions, sir, from
Yeah, yeah,
participants?
yeah, yeah. It's my request to the participants if they have any question. Very good. Okay, so I think we have no other question from the
Thank you, sir.
participant. So thank you so much, sir, for your presentation. Now I
Thank you,
will,
sir. Thank you,
yeah,
sir.
yeah. Thank you, sir. Thank you so much. Thank you so much. Now I will request Miss Songita Mondol to present her presentation. The topic is weed warriors of nature using allelochemicals to create bioherbicide solutions. Ma'am, are you there? Hello, Miss Songita Mondol, are you there? I think Miss Songita Mondol is not present there. So I will request the next participant, Miss Prerona Bhuiya. Miss Prerona Bhuiya, are you present there? Hello, am I audible? Um, Dr.
Yes, you
Prashant
are
Saha,
audible.
yeah, I think
Maybe they are
uh,
not present.
Dr. Prerona Bhuiya is not present there. Okay,
Shall we
so
move to the next?
I will move to the next participant, Dr. Probal Das. Are you present there, sir? Hello, Dr. Probal Das, are you present there? Yes. Hello, sir. You are not audible, sir.
Hello.
Yes, sir. Now you are audible. Yes, sir.
Okay, sir, may I present?
Yeah. So we have Dr. Provoldas with us, and his topic is calcium attenuates the
Yes.
negative effects of chromium stress by modulating antioxidant defense system in black ground, Vinca mungo paper, and seedlings. So I will request sir to present his slide.
Okay, sir, I'm present.
Okay.
Sir, am I audible?
Yeah, you're audible and your slides are visible. Kindly move it to the slideshow mode.
Okay, okay, sir.
Yeah. Now
Is
it
it
is
okay
clearly
now?
visible. You can start now. Yes.
Good afternoon, everybody. My name is Dr. Prabal Dash, Assistant Professor, Department of Botany, Tamranitya Mahavidyalaya. Today I'm here to present my topic regarding calcium attenuates the negative effects of chromium stress by modulating antioxidant defense system in black gram Vignamungo seedlings. As you all know that the accumulation of heavy metal in the environment due to modern industrial development is among the most serious pollutions around the world. At present, heavy metal pollution mainly includes mercury, cadmium, lead, and chromium, as well as arsenic. Numerous studies have reported that high concentrations of chromium compounds contaminate the soil and hindered the germination and growth of many crops, including legumes. Chromium is the seventh element on Earth and has various valence states. Among them, the most prevalent one is hexavalent and trivalent one. The trivalent one is to some extent less toxic than the hexavalent one. And chromium accumulated in plant enters the body through the food chain, causing dermatitis, bronchitis, tuberculosis, and so on. It has been seen that the hexavalent chromium is 100 times more toxic than that of trivalent chromium. In this slide, I have shown the effect of chromium toxicity on plants in a nutshell. You can see the effect has been distributed in four categories, starting from morphological effect, physiological effect, biochemical effect, and molecular effect. In molecular effect, it damages the DNA, arrests the cell cycle, as well as alters the transcription and translational level of the proteins. And in biochemical effect, it alters different raw scavenging pathway, which is the main objective of my study. I will later on show in slides. Enzyme activity, lipid peroxidation, photosynthesis, and so on. And as well as morphologically, you can see it results in stunted growth of the seedlings, leaf growth, chlorosis, and so on. In my study, I have tried to ameliorate this chromium-induced toxicity in Vigna mungo seedlings by application of calcium. Why I have chosen calcium? Because calcium is being a macronutrient plays a key part in plant growth regulation. It has a crucial role in controlling the structure and function of cell membrane. Calcium, by binding to phospholipids, upholds the structure of lipid bilayers and thus providing structural integrity to plasma membrane. It has been seen that calcium can mitigate different heavy metal toxicity, including cadmium, in different other legumes and rice. And also, calcium alleviates cadmium-induced oxidative stress in plants by scavenging reactive oxygen species, increasing the antioxidant level. So it has already been reported. So I am trying to see whether this effect is there in my seedlings or not. So now coming to my objective of my study, first one is to characterize influence of chromium on black gram seedlings using biochemical parameters. Number two, amelioration of chromium toxicity in the test seedlings by calcium. Number three, to assess the effect of chromium stress on the level of stress markers like proline, MgA, peroxide in my test seedling, that is Vignamungo. And last one is to isolate and study the role of different antioxidant enzymes like SOD catalase and different metabolites like TPC and phenols during chromium toxicity. Now I describing my material methods in a nutshell. After procuring the seed, we have disinfected with 5% sodium hypochlorite, and after germination in the petri dish, we have given 3 doses of potassium chromate like 150 micromole, 300 micromole, and 600 micromole concentration for 21 days with or without 10 millimolar calcium chloride. And after 21 days, the root and shoot are separated, and these were used for further analysis. First, we have measured the pigment content like chlorophyll A and chlorophyll B according to our norm. Second, we have measured the sugar content like total sugar and reducing sugar according to the protocol mentioned in the slide. Third one is different non-enzymatic antioxidants like total phenol and flavonoids. Fourth one is different enzymatic antioxidant like superoxide dismutase (SOD) and catalase. Last, we have measured different stress markers like H2O content, MDA, malondialdehyde, as well as proline content according to the protocol mentioned. We have estimated the protein according to Lowry, and in statistical analysis, we have undergone one-way analysis of variance to compare the significance among concentrations of chromium in presence or absence of calcium independently. Going to the result section, in the first slide, I have shown the effect of chromium and calcium on growth and total chlorophyll. It is very much clear from the graph that there is a gradual decline in root and shoot growth as well as total chlorophyll content upon a dose-dependent manner of chromium application, and the root were very much susceptible to chromium stress, and joint application of chromium along with calcium chloride ameliorated this growth in case of both these cultivars. In the next slide, I have shown the effect of calcium and chromium on chlorophyll A and chlorophyll B. Like total chlorophyll, you can see here also chlorophyll A and chlorophyll B content decreased linearly, and the amount of decrease was maximum in 600 micromole chromium treatment. And when calcium was jointly applied at chromium, the level of decline is to some extent reversed. In the next slide, I have shown the effect of chromium and calcium on sugar contents. We have measured two different types of sugars, that's total soluble sugar and reducing sugar. Since photosynthetic machinery is hampering, so here we can see the total soluble sugar content as well as reducing sugar content both decreased linearly in a dose dependent manner. Joint application of calcium along with chromium reduced this level of decline in my test seedlings in both root and shoot. Next is effect on total phenolic content and flavonoids. Like the sugar contents, also total phenolic contents and flavonoid contents decrease linearly. And upon application of calcium chloride along with chromium, you can see the amount of decline is to some extent reversed. That means the amount has been increased compared to the only chromium treated seedlings. Now I have measured two different enzymes of the raw scavenging pathway. First one is SOD and second one is catalase. You can see the activity of catalase enzyme declined in a dose-dependent manner, and whereas SOD activity increased linearly. And when calcium was jointly applied with chromium, you can see the level of decline in catalase activity is to some extent narrowed down, whereas there was a further enhancement of SOD activity, noticing the upregulation of raw scavenging pathway. Now I have shown the effect of chromium and calcium on H2O2 and MDA. Both are very important stress markers. In both the cases, you can see in both root and shoot of the seedlings, diagram seedlings, you can see there is a accumulation of H2O2 and MDA content upon exposure to chromium stress, and joint application of chromium along with calcium narrowed down this accumulation of both the stress markers. Last stress marker which I have measured is proline, like H2O2 and MDA. Proline contents also increase linearly, and when we have applied the calcium chloride, you can see the accumulation of proline is to some extent decreased. So from all of this assay, we can conclude that 10 millimolar calcium along with chromium shows its beneficial effect to mitigate the chromium-induced toxicity in my test seedlings. My different parameters assessed like chlorophyll, sugar, phenol levels decreased upon exposure to different levels of chromium, whereas when calcium was jointly applied with different doses of chromium, this level increased compared to the only chromium stress. So we can conclude here that co-application of calcium chloride with chromium can mitigate these effects, and we can use this in our field application in the near future. I would like to thank Department of Botany, Tamralipta Mahavidyalaya for their instrumentational and administrative support. And now, thank you, sir.
Thank you so much, sir, for your nice presentation. So I would like to request our participants to see if they have any question. Dr. Ashok Bhattacharya has raised his hand. So, sir, you have any question? So, Dr. Sandeepan Ray has some question, and after that, we'll move to Dr. Prashant Shah. Dr. Sandeepan Ray, kindly ask.
Dr. Probal, are you here? I guess he left the meeting.
I guess he left the meeting somehow or some technical problems up there. So,
Dr. Prabal Dash, no, he left the meeting.
uh, okay, yeah, Dr. Prabhudas is here. So you can ask Dr. Sandipana, you can ask your
Yeah,
question.
Dr. Prabal Dash.
Yes, yes, sir, please ask.
Yeah, am I audible to you?
Yes, audible.
Hello.
Yes, you're audible. Please ask.
Yeah. I have a very simple and a quick question. That have you seen the effect on the yield? Because at the end of the day, what we need is the yield, right?
Yes, sir. Yes,
So what
I
the
can't
effect?
show the
Is
yield,
it?
but it is very primary work, and we have done the hydroponics,
Mhm.
and we want to see it in the near future for the pot experiment and in the field culture also. Then we
Yes.
will go for the yield. Because I have
Means
already mentioned that it's 21 days condition, the seedling stage,
yeah,
and in seedling
seedlings.
stage,
And unless
is
or until you are going to the yield,
yes, we
means
cannot conclude.
then yeah, you can conclude anything that chromium or calcium,
Interaction of chromium and calcium
they're
on the yield.
together. Yeah,
Definitely.
they're increasing the yield because at the end of the day we need the product.
Products.
Whatever the
From the
vegetative
product
stage
we need
it
it
will
actually.
be, what the effect in the vegetative stage, but the yield should be same
Measure.
or high. Yeah,
Yes.
that's it. Thank you.
Thank you.
Thank you so much, Dr. Ray. So I will move towards Dr. Prashanto Shah if you have any questions. Sir, please.
Okay, Dr. Probal Das. Okay,
Yes, sir.
so first of all, it was a very good presentation from my end. I just want to know two fundamental things. I mean, why did you selected Vigna as your stressor of
Since it can be grown easily in hydroponics and it is a very economically important crop, and it has been seen that this chromium contamination is actually decrease the yield of Vigna mungo, this black gram. And in West Bengal, we actually use it for dietary nitrogen content. So it is very
Huh?
economically as well as nutritionally important.
I mean, the question was whether chromium toxicity
Hmm.
is the chief stressor of Vigna or any other parameters
No,
like
there are different stressors also, but among the commercial effluent, like cadmium, chromium is
mhm,
also a very important stressors for abiotic
right.
stress in case of Vietnam.
And what in terms of actually this relates to further questions of the key interest by
You are not
chromium
audible.
toxicity. I think rice, wheat, they would they are stressed to a greater extent, isn't it?
Actually, sir, you are not audible to me for technical error now.
Yeah, your voice is
Yeah,
breaking, Dr.
Okay.
Prashant.
yeah, your voice is very
Okay,
low.
okay. Okay, then I'll just end it over here. I mean, the question was whether means uh, other crops like rice and wheat, they are I mean they challenged to a greater extent, I suppose, by chromium toxicity, isn't
Definitely,
it?
rice is greatly extent challenge to chromium stress.
Mm,
There are lots of papers and researchers are there regarding chromium toxicity and rice seedlings because rice is the major food crop for India and as well as West Bengal.
exactly. And lastly, regarding as you mentioned that you went for hydroponic treatment.
Yes.
So did you use any solution, nutrient
Yes,
solution
we
like the
have used
Hoagland
the modified Hoagland solution.
modified Hoagland solution?
Modified
Okay.
Hoagland.
Okay. Thank you. I liked your
Thank
presentation very much. Thank
you.
you.
Okay.
Thank you so much, Dr. Prashanto Shah. So I will ask Dr. Ashok Bhattacharya sir if you have any question. Sir has
No,
raised.
actually, I don't have any question. I like her. I liked his lecture because objective was very clear to me, and that's why just it was my like, no question. One suggestion I think I'm giving to Dr. Proval. He has presented the work is very nicely, and work is in preliminary, the first stage one, I think, and that it will be enriched more in future because it is the very interesting work, the cadmium, nickel, and the chromium toxicity, and that those are affecting as the stressors molecule. So in future, I think that those stressors will be characterized biochemically by different. Say for example, the proline is estimated only, but it will be the characterized under different conditions. The what level of the proline is influential, and I think that in future it will be correlated with ROS. That means the stress when it abiotic stress is related, there is the reactive oxygen species level is also the one important thing. That's why I am suggesting that in future Dr. Pubal will do it. The correlation with the ROS, that means the reactive oxygen species activity along with the different concentrations of the stressors and the molecule stressors molecule, the characterizations of the stressors molecules, like some other related work. It is very nice work, and the plant physiological aspect, advanced plant physiology, particularly in the stress physiology aspect, it is very nice. I would like to thank you a lot to Dr. Proval for his bright career in this particular endeavor.
Yes.
Thank you, Dr. Proval. Okay, okay.
Thank you so much, Dr. Bhattacharya, for your kind suggestions. So, thank you so much, Dr. Probal Das, for your presentation. And now I will move towards Dr. Devashish Bandyopadhyay to kindly present his presentation. And the topic is sulfur mitigates cadmium toxicity in mung bean seedling by altering pigment and antioxidant levels. Sir, are you there?
Yes, sir, I am audible. I am in here.
Okay, okay, sir. Okay, sir. So it's a kindly. Do you want to present by yourself or we can share here?
Sorry, just a minute, sir. Just
Okay,
just a minute, sir.
okay, okay, okay,
I'm
okay,
not audible properly. Okay.
okay, okay.
Hello, sir. Just
Yeah, yes, you're audible.
hello.
Yes, sir, you're audible, sir, and the screen is visible clearly, and the slides are also moving.
Hello.
Yes, sir.
Yes, sir.
Hello.
I'm audible. Okay. Okay.
Yeah, yeah. Your screens are visible also, and slides are moving also.
Okay. Uh, Good afternoon to all the respected teachers. Uh, I am Dr. Devashish Bandyopadhyay, Assistant Professor, Tamiluttu Mahavidyalaya. Today I am saying about the sulfur mitigates cadmium toxicity in movement seedling by altering the pigment and antioxidant levels. Cadmium is a highly toxic to living organisms and humans. It is a heavy metal and development in agriculture and industry it has been calculated in the higher concentration in cadmium in agricultural soil. Cadmium coming into the agricultural soil from different sources, mainly from fossil fuel combustion, different type of industrial wastewater, different type of treatments, different pesticides. From this, it comes to the soil by irrigation. And cadmium toxicity affect the plant by inhibiting the fixation of the carbon and decreasing the chlorophyll content and photosynthetic activity. Uh, the effect of cadmium toxicity can differently uh, differently induce the plant stress uh, by by degrade its photosynthetic activity, grow different type of enzymatic activity, and it also decrease the crop yields. This cadmium stress can be eliminated or minimized by the effect of sulfur. Sulfur is a essential element for plant which involve the biosynthesis of sulfur containing compound such as different type of amino acid that is cysteine, methionine, etc. And positive effect of sulfur fertilizer on the ability the heavy metal toxicity in crop plants have been extensively investigated. Sulfur supply retain the heavy metal uptake through enhancing the iron plate formation on the root surface and which uh, try to stop the cadmium absorption and and ultimately the sulfur and sulfur supply limit the heavy metal translocation to soot and increase plant tolerance to stress to regulating the biosynthesis of thiol compounds. Now come to the objectives. We characterize the influence of cadmium in mood bin seedling using the biochemical parameter related to growth and metabolism. and minimizing the cadmium toxicity in the test seedling by potassium sulfate. Next, to assess the effect of cadmium stress on the level of stress markers like as proline, MDA, peroxidase, and pigment in test seedling. To isolate the study of the role of different antioxidant enzyme SOD, catalase, and different type of metabolites, that is the TPC and flavonoids have been studied. Now come to the methods and method. In the preliminary stage, moving seed were disinfected in 5% sodium hypochlorite solution, followed by transfer of petri dish containing water, followed by incubation at 30 degrees centigrade in dark for germination. The germination seeds were transferred to petri dish and lined with the filter paper containing different test solution of cadmium chloride in different concentration, 25 micromole, 50 and 100 micromole concentration, with or without 5 millimole potassium sulfate concentration. And for 21 days, and after that the root and shoot sample were collected and it was treated as the study testing materials. Uh, we have studying the different parameter for stressing. There is a pigment content, estimation of sugar, estimation of total phenols and flavonoids, assay of enzymatic antioxidant, assess test different type of marker, H2O2, MDA, proline content, and protein estimation. Now come to the result. Cadmium treatment diminished the growth and inhibition was greater in root compared to that of shoot compared to control. Surface amendment, sulfur amendment in the cadmium stress sample also inhibited root as well as shoot growth, but to a lesser extent compared to the only cadmium treatment. Total chlorophyll content also measured, and total chlorophyll content also reduced the test cultivar under the 25, 50 and 100 micromole cadmium treatment and respectively with respect to control. On the other hand, in sulfur supplement seedling, decrease in total chlorophyll content narrowed down compared to control and was greater than the corresponding cadmium treated sets. Chlorophyll A and chlorophyll B also assayed. The chlorophyll A and chlorophyll B content both decrease in the cadmium challenge seedling in comparison to control. Sulfur supplementation along with cadmium narrowed down the decrease in chlorophyll A and B level under similar treatment. However, chlorophyll A and B level in the sulfate supplemented seedling were greater than the individual cadmium treated seedling. Carotenoids and anthocyanin also acid. Like chlorophyll content, carotenoids and anthocyanin content both decrease in the cadmium channel seedling in comparison to control. Sulfate supplementation along with cadmium narrowed down the decrease in chlorophyll A and B level under similar treatment. However, carotenoids and anthocyanin in the sulfate supplement seedling were greater than the individual cadmium treated seedling. Soluble sugar and non-reducing sugar also assayed. The total soluble sugar and reducing sugar content also reduced the test cultivar under 25, 50 and 100 micromole cadmium treatment respectively with respect to control. On the other hand, the sulfur supplement seedling decreased in total soluble sugar and reducing sugar content, narrowed down in root and shoot compared to control, and was greater than the corresponding cadmium treated cells. The cadmium effect also shown in the phenolic content and flavonoid content. The total phenolic content and flavonoid content are reduced in the test cultivar under cadmium stress. On the other hand, in sulfur supplemented seedling, decrease in total phenolic content and flavonoids narrowed down in root and shoot compared to control and was greater than the corresponding cadmium stress treated set. Sorry. And in case of SOD, superoxide dismutase activity increase the root and shoot respectively at 25, 50, and 100 micromole concentration, cadmium concentration. And in respect to control, and supplementation of sulfate conjugation with cadmium further increase the enzyme activity in root and shoot under same treatment. In case of catalase, cadmium treatment decrease and catalase activity in the test cultivar corresponding over control. The decrease was more prominent in root than shoot, and administration of sulfur together with cadmium narrowed down the level of decline in catalase activity in root and shoot over control, which again greater than the cadmium treated samples. Hydrogen peroxide and MDA content significantly increased with cadmium treatment in seedling with respect to control. Administration of cadmium along with sulfate also increased hydrogen peroxide and MDA content, but to a lesser extent compared to individual cadmium treatment. In case of proline, like hydrogen peroxide and MDA content, proline content significantly increased with cadmium treatment in the seedling with respect to control. And administration of cadmium along with sulfate also increased proline level, but to lesser extent compared to individual cadmium treatment. In conclusion, cadmium application reduce root and shoot length in moving seedlings. However, the growth inhibition in moving seedling caused by cadmium stress was ameliorated by the application of high-valent potassium sulfate along cadmium, indicating the minimum effect of K2SO4 in cadmium tolerance process. Total chlorophyll, chlorophyll A, chlorophyll B, carotenoid, and anthocyanin content showed a gradual decline in test seedling. Co-application of 5 millimole potassium sulfate with cadmium chloride reduced the rate of chlorophyll degradation compared to only cadmium treated plants. Cadmium toxicity adversely adversely uh, altered the activities of antioxidant enzyme like a superoxide dismutase and catalase. Cadmium stress also enhances the level of oxidative stress marker like as proline, hydrogen peroxide, MDA in both root and shoot of the seed. Co-application of potassium sulfate and cadmium chloride was able to overcome all such adverse effects to some extent, leading to better growth of the test seedling. I like to thank my college, Tamralipto Mahavidyalaya, and especially to the Central Laboratory for constant support uh, physically and administratively. Thank you.
Thank you so much, sir, for your nice presentation. So I will request the participants if they have any question. You can ask if you have any questions, dear participants. Okay, so I think for dear participants have don't have any question. So in that case, I will move towards our next participant, Dr. Osmita Pal. Ma'am, are you there?
Yes, I'm
And
here.
yeah, yeah. So Dr. Osmita Pal is presenting us her presentation, and her topic is Tiliacola alkaloid potency in cancer phytomedicine. So, ma'am, do you want us to present your slide, or you will share by yourself?
I'll share by myself. Let me just try it
Okay,
first.
okay, ma'am. Thank
Is it visible?
Yes, ma'am, it is visible. It is clearly visible,
Okay.
and it is moving also.
Okay. Thank you. So I'll begin my talk now. So good
Okay,
afternoon, everyone. I'm Dr. Ashmita Pal, and I'm currently working as an assistant professor in Department of Botany in Government General Degree College, Narayangarh, where I've been recently posted. And today I'll be presenting a part of my doctoral research work, which I have carried out as a CSIR UGC Research Fellow in Department of Botany, University of Calcutta. But before I begin with my work, a very alarming situation I would like to present to you about cervical cancer in India, where 1 in every 50 Indian women is predicted to develop cervical cancer, and India accounts for around one third of the total global cervical cancer deaths, which I guess is huge. And the major problem behind that, or the major stakeholder behind it, is the development of drug resistance in cancer cells. That is, the already available chemotherapeutic drugs, the cells are becoming resistant to them, and we need to find a solution to it. And talking about the solution, our mother nature is the one who we always come to. And right, starting from an early stone age to the present day doctors, everyone has been relying on the use of plant based compounds or the plant based extracts, which scientifically we term as phytotherapeutics, for the treatment of different kinds of diseases, but I'll be talking about particularly cancer based diseases here. And they are based majorly, we rely on them due to their multi targeting ability, broad availability, and low toxicity. Many anti-cancer drugs have been already developed from them: podophylotoxin, vincristine, camptothecin, and taxol. But we do need novel sources as well, because the cells are very smart and they become resistant very easily. So novel sources of medicines are also very important. So talking about that, my research work was started from the forests of Jhargram, from where initially 4 plant specimens like Tiliacora racemosa, Clistanthus collinus, Eupatorium odoratum, and Sida acuta, the leaves from these plants were phytochemically extracted in 4 different kinds of solvents, and they were tested for their anti-cancer potential in cervical cancer cell line, where we observed that Tiliacora racemosa methanol extract, which I'll be referring to as TRM from now onwards, showed the most potent cytotoxic effect with the lowest IC50 dosage, because of which this plant extract was selected for further studies. And we noted that its anti-cancer potential was most pronounced in Shiha cell line, which is infected with human papilloma virus 16, in comparison to Hela cell line, which is infected with HPV 18, and C33A, which is not infected with any HPV. But the most dominating effect was seen in Shiha cells, and therefore we carried out all other studies in this Shiha cell line. And but before that, we also observed that there was around 6 times window of a TRM cytotoxicity in a non-cancerous HEC293 cell line, which is a kidney cell line. That is where we see that it is selectively cytotoxic for cancer cells only. And this anti-cancer potential was further proved using clonogenic assay and wound healing assay, where the clone forming potential and the wound healing property of these cancer cells were greatly reduced in presence of TRM. So, to find out how this TRM was actually inducing or exerting its cytotoxic effect, we firstly note that it was able to generate reactive oxygen species or oxidative stress in cells by around 12-fold in response to the maximum concentration that was used, and it was also successfully scavenged by NAC. By NAC, I mean N-acetyl L-cysteine, which is a very well-known antioxidant, when we conclude that TRM is actually working by inducing oxidative stress in these cancer cells. So, continuing with this oxidative stress, along with the flow cytometric studies, microscopic studies also showed that there was a continuous increase in ROS production in response to the increasing dosages of TRM, and the use of NAC treatment was able to successfully scavenge it as well. So, in relation to the redox balance only, we also observed that the TRM was able to generate nitrite production, that is extracellular nitrite production, because of which probably the malondialdehyde content of the cells were also getting increased, that is, the lipids were getting peroxidized, and the cellular antioxidants of the cells were greatly reduced, that is, superoxide dismutase by around 6-fold, catalase by around 70 folds, and glutathione by around 32 fold. This is how probably TRM is acting, that is inducing the formation of reactive oxygen and reactive nitrogen species, and by reducing the cellular antioxidant level. So because the oxidative stress was so huge, we needed to find out what was the condition of the cellular constituents, that is DNA, protein, and etc. And we find that TRM was actually inducing DNA damage. This comet assay clearly shows that in response to increasing TRM dosages, there was successive increase in the DNA damage of the cells. There is around 22-fold increase in DNA damage was noted, and in association with that also we find that the DNA damage was double stranded in nature, which we studied by studying the gamma H2X foci formation under a confocal microscope. The gamma H2X foci can be clearly seen as green fluorescent specks in the TRM treated cells, which we do not observe in the control cells, which show that the DNA damage is double stranded in nature. And in relation to that, many proteins were studied for their expression status, where we find that all of the proteins clearly prove that the DNA were getting damaged, and other proteins like CHK2, which is known for inducing the further downstream cascades, were also getting induced. Part 1 is a molecule which we also studied by flow cytometry and found to be induced by around 5-fold in response to TRM, is another protein which is related to DNA damage. So till now we observed that there was a double-stranded DNA break, which is probably being sensed by molecules like PARP1, gamma H2X, DFF45, and ATM. They were inducing the expression of checkpoint kinase 2 protein and P53 in its phosphorylated form, which was inducing the expression of P21. So now we wanted to see if there was any changes in the cell cycle status of the cells, and we find that the cells were getting arrested in the G2M phase. That is, there was around 30% cell population in G2M phase, which in control is nearly to 18%. So why was it happening? We found that the cyclin B1 molecule, which is actually a checkpoint detector at the G2M phase, was getting greatly reduced. So decrease in this cyclin B1 pool was what was resulting in induction of this G2M phase arrest, which was actually a result of the increase in probably the P21 protein, which I showed in the previous slide. So now in association to this, we wanted to see what kind of cell death was being induced, and we saw that the cells were actually embracing apoptotic type of cell death. That is, we find that 84% of cells were in apoptotic stage, early and late combined, which was greatly then reversed by using ZBAD FMK, which is an apoptosis inhibitor. Now, in relation to this apoptosis, we wanted to see that what kind of pathway was being induced, and we did a proteome profiler assay where we found that pro-apoptotic proteins were upregulated, anti-apoptotic proteins were downregulated, extrinsic apoptosis proteins were again upregulated, and apoptosis inhibitors were down, DNA damage proteins were up, and there were certain changes both up and down in heat shock proteins. So, and the executioner molecules, that is the last stage molecules of apoptosis, are known as caspase molecules. The caspase 3 is a very important one, which was also greatly increased, which again then successfully reversed by using ZVAD FMK, which I stated as an apoptosis inhibitor. So TRM induces apoptotic cell death. These are some more evidences of it. These are acridine orange ETBR stained cells, which also show how the cells are embracing apoptotic cell death. These are the caspase molecules, which are the last stage executioner molecules, and APAF1 molecule, which was getting upregulated, and these caspase molecules are getting cleaved, showing that apoptosis was being induced in response to TRM, which is probably a result of the DNA break and the G2M phase arrest. So, and in association to that, we had also needed to see that what was the status of the mitochondria in the cell, because mitochondria is a very important member, and we find that the mitochondrial membrane potential, that is MOMP, was greatly reduced. That is, the cells in the green phase here, that we have performed a JC1 staining, where the JC1 monomer, which appears green, was successfully increasing, and the JC1 aggregates, which appear red, were successfully decreasing, which clearly proves that there was a decrease in the mitochondrial membrane potential, which was further studied by rhodamine staining in microscopy, which also stated the same thing. And the mitochondrial membrane potentials, that is the mitochondrial membrane proteins which are associated in the mitochondrial membrane, like BAX, it's a pro-apoptotic protein, was getting upregulated. BCL2, an anti-apoptotic protein, it was downregulated, and BEAT, which is also found to be in truncated stage, were all again clearly showing that how apoptosis was happening. That is, dysregulation of this BAX BCL2 status is another very important marker of apoptotic cell death. So another side observation was that TRM also inhibits PI3K AKT NF kappa beta pathway, a well-known pathway. It was also probably culminating in apoptosis. So talking about all of this, we needed to see now that these all of these effects, cytotoxic effects, were result of actually exactly which point. And we find that NAC, or use of a N-acetyl L-cysteine, which can actually reverse the oxidative stress, was able to reverse the cytotoxic effect, the induced apoptosis, as well as the induced DNA damage, from which we conclude that TRM induced cytotoxicity is a direct result of oxidative stress. And from there, the mode of action that we have been able to draw is that TRM, the one shown in a green box above, induces oxidative stress in the form of ROS and NO species, and the antioxidants also go down, which results in double-stranded DNA break, which by a CHK2 kinase pathway induces G2M phase arrest through cyclin B1 inhibition, along with an induction in the expression of extrinsic apoptosis and intrinsic apoptosis proteins, causing loss of mitochondrial membrane potential, inducing the expression of caspase molecules, which ultimately culminate in apoptosis. And there is another side pathway which I stated, that inhibition of PI3K AKT pathway, which I am not going in details here, was also inducing apoptosis. So
Kindly conclude, ma'am.
yes, I'm on the last only.
Okay,
So
okay.
this methanol extract that I have been showing was actually rich in alkaloid molecules. The LCMS analysis show us that it was rich in many kind of alkaloid molecules, particularly teliacodenine 2' N oxide, and these leaves contain a very great amount of alkaloid content, and the alkaloids which were isolated was identified as a probable bioactive compound in the Shiha cells with an IC50 of 3 microgram, and in the non-cancerous cell line again it gave us a similar cytotoxicity window of 6 times. So by that I come to the conclusion, and we conclude that this teliacoid alkaloids, chiefly teliacodenine, can serve as an excellent drug candidate, particularly for cervical cancer, and we have been able to publish this in the Journal of Ethnopharmacology. And this is my acknowledgement slide where I acknowledge my PhD supervisor, Professor Rita Kundu ma'am from Department of Botany, University of Calcutta, and my greatly enthusiastic, wonderful team, the photo that I've shown here, as well as the Center for Research in Nanoscience and Nanotechnology, University of Calcutta, UGC for my fellowship, and last but not the least, definitely Department of Botany, Durgapur Government College, and DBT for giving me this wonderful opportunity to present my work in front of everyone. I thank everyone for a patience hearing, and I greatly welcome any kind of questions. Thank you.
Thank you so much, ma'am, for your beautiful and excellent research work. So I will request our participants if they have any question, they can ask.
Sure.
It was a flawless work. So let's see if they have any question or not.
Okay.
I think the participants have no other question from their side.
Yeah.
So thank you so much, ma'am, for your beautiful presentation.
Thank you. Thank you, everyone.
Okay. So now let's move towards the next presentation, that is by Mr. Deepan Bandyopadhyay. His topic is ethnobotany and medicinal uses of selected plant species in West Bengal belonging to Lamiaceae family. An updated review. So Mr. Deepan Bandyopadhyay, are you there?
Yes, sir. Am I audible?
Yeah, you are clearly audible. Kindly present your slide.
Okay, I will share my slide.
Sure.
Actually, sir,
So do you want us to share the slide from our side?
sir, kindly give me 1 minute, please.
Okay, okay, sure.
I will try. Is it visible, sir?
Yes, yes, clearly visible. You can start.
Okay, I will
Kindly, yeah,
leave it
kindly
like
move it to the slideshow mode,
right.
sir,
So,
please.
uh,
Yeah, yeah, yeah.
it in right. So, na.
Yeah, yeah, yeah. It is now okay.
So,
Fine, you can start, sir.
first of all, I am very much thankful for the organizing committee of Durgapur Government College for providing me these facilities. So, without delay, I would start to my presentation, that is ethnobotany and medicinal uses of selected plant species in West Bengal belonging to Rabiati family, an updated review. So, the Rabiati family, presently known as Lamiaceae, derived from the type genus Lamium. Lamiaceae, commonly known as the mint family, is phylogenetically considered to be one of the most highly evolved among the dicotyledonous family. So we know the most advanced family is the Compositae or Asteraceae among the dicot, and the research study revealed that Lamiaceae is the second advanced family in the dicot. So, what are the objectives of the study? First of all, identification of medicinal plants of Tabiati and its distribution in West Bengal to unveil the importance and significance of medicinal and aromatic plants in the treatment of common diseases, to increase the dependence on the herbal treatment instead of allopathic treatment, as there were too much side effects. And the fourth one is to promote the consciousness and the awareness on the conservation of medicinal plants. Number five, processing, that is too much vital, that is processing of herbal products and phytoresources in a scientific way for its commercialization and engagement of younger generation for the development of rural economy. And number six, utilization of traditional knowledge of indigenous groups on herbal medicine and its application with proper uh, method, and secure the sustainable man-plant relationship for the development of human society. So, what is ethnobotany? So, it is the definition, that is the sum up of father of Indian ethnobotany, Professor S.K. Join, who defines the branch of biological science dealing with the direct and sustainable man-plant relationship, including food, fodder, medical practices, decorative, spicy, aromatic, and others uses. So the now question is arise, why the Rabiati? So the Rabiati family have some unique characteristics. That is aroma, quadrangular stem, fascicular inflorescence, and schizocarpic fruits. Actually, schizocarpic fruits are the broad perspectives, and mainly the Rabiati fruit types is the carcerule type of fruit. This review documents 15 selected medicinal plants of Rabiati found in West Bengal, emphasizing their vernacular names, plant parts, mode of administrations, and the disease treated. So, Osimum tenuiflorum, that is commonly known as Krishna tulsi, plant parts are used in leaves and seeds, mode of administration, infusion, decoction, fresh juice, medicinal uses, antimicrobial, anti-inflammatory, and it is widely used in common cold, cough, fever, respiratory infection, etc. Osimum basilicum, here plant parts used leaves, seeds, and mode of administration essential oil, digestive problem, antimicrobial and disease treated headache, indigestion, etc. And the most important plant, that is Leonidus sibiricus, commonly known as Doctodrone. So aerial plant parts are used, decoction, infusion, uterine, tonics, sedative, etc. And this photograph is taken from the Howrah district of West Bengal. And the next is the Leucas aspera, that is cedron. The plant parts used leaves, flowers, mode of administration, decoction, paste, antipyretic, expectorant, cough, skin problems, etc. etc. disorders. and that is the another important plant, 50 violence. Here the plant parts, leaves, seeds are also used, antimicrobial, analgesic, etc. etc. And the next vital plant is Anisomelis indica. It is a huge ethnomedicinal importance in fever, in digestion and medicinal use. It is also used as antipyretic and carminative. So this is the another species of Anisomelis, that is Anisomelis ovata. In a mode of administration, infusion, paste, leaves are used, skin infection, digestions are created. So this plant, Vitex gigando, it is commonly known as Hisinda. But I want to say more another point, that Vitex gigando is placed in Herbaceae family under the Benthamucca system of classification. But the new APG, angiosperm phylogeny group, this Vitex is replaced in Tabiati. It is highly medicinal plant and widely used in arthritis, fevers, leaves, roots, and seeds are these plant parts are used. Pterodendron infortunatum, this is commonly known as Ketu or fat flower, leaves and roots used, mode of administration, paste, decoction, hepatoprotective, etc. Liver issues, skin problems are widely used, and I want to mention this plant is widely found in Nadia district, and this photograph is